Thanks, Jenny. Very kind. So, you usually come up here and thank the organizers, so that'd be a little self-serving in my case. More of the story is the way to get yourself on the agenda is make the agenda. And I feel like I have the home field advantage because if I get any difficult questions, I've got all my colleagues here that can help me answer them. So you're only 87 slides away from lunch. No, I'm just kidding. It's not that many, but we'll roll through here and talk a little bit about a little bit about plasmids, which are near and dear to our heart, and how we're moving from bench to bedside. So if you look up bench to bedside on the internet, really it's describing how we get results from the laboratory and to develop new ways to treat patients. So, we start in the laboratory and we eventually want to get here and make people feel better. In the interim, there's quite a bit of work to be done and challenges and such, so we want to be able to, meet those challenges and get things even faster, safer to patients. So really today, when it comes down to it, sponsors are ultimately responsible for their products. So, we want to be helpful to people and contribute to the industry and really provide options for people. What are the options? Help think about when to use these options and even importantly, how do we move faster through these development processes from the bench to the bedside? So when you think about plasmids at the bench and the bedside, what are the real differences? So, each of these products that we sell has a certificate of analysis, can be sequenced, so, they have a lot of similarities. And when you go through the certificate of analysis, we'll go through sort of the anatomy of a C of A, you find out they're, usually more alike than different. So a lot of these assays and specifications are consistent from the bench to the bedside. So we don't see cases where, like if you're working with a certain amount of supercoiled percentage and such, that doesn't sort of get tighter as you move into the bedside. It's other aspects that we'll talk about that change on that. So that's actually a good thing because we can have that consistency as we move closer to the bedside. So what are the differences? Sometimes we have additional standard tests. So like in this case, we have the sterility and the mycoplasma, and those are really add-on tests. Again, the specs aren't necessarily changing as we move from the bench to the bedside, where we're adding more tests. And that's a good thing because some of these specifications like supercoiled and endotoxin, will drive price. So the fact that they're consistent throughout the journey from the bench to the bedside is a good thing and it's helpful. So these C of A's that I'm showing here are actually from a product we sell, as Jenny referred to. We have a Ph.D. X80, which is a helper plasmid that we've developed along with partners at IGEN. So it's a really good example of something that we can look at and talk about this journey from the bench to the bedside. So this is not from the C of A, but this is a list that we can provide people when they are looking at clinical grade that are additional optional tests that we come across and some clients are interested in. So a lot of them we use outsourced vendors to execute, and we do custom assay development, and these are different for different situations, and we'll talk a little bit about that and later in the talk, things like particle matter and injections, when might you do that, but there are different uses for plasmids, and these tests support those various uses. And the other difference is the QC service level. So in the research grade, the sort of purpose is to create something that is very quick turnaround and low cost. So we can't burden that product line with extra qualification for assays and things like that. But when we move toward the bedside, we need that. So you'll notice on the C of A's, we refer to certain SOPs for all of these assays that we do, or we use qualified external vendors. So that's a difference between these different service levels as you move closer to patients. And then, of course, we'll talk a little bit more about the production service level. So in the case of the research grade, you're going to get a certificate of analysis that specifies it was made under research grade conditions, and in the case of what we show here, GMP source conditions, and that says something about how the material has been manufactured, the quality systems, the facilities, and we'll talk about those things. But I think the point being is sort of the stuff in the vial, is largely the same if you're looking at the sequence and these other specifications for these two different products. So when you think about the basic process in both these situations, it is the same whether you're making things for research grade or for clinical use. We transform bacteria, we grow, so this is a 10-liter fermenter that's in our research grade facility. We harvest, so we have centrifuges all over the facilities, both in research grade and in the clinical areas. Lysis, so we talked a little bit in the past about larger scales, and this is one of the larger scale lysises that we've done. We use chromatography, so just a matter of the size of columns. It may be a different number of columns depending on what the specifications we're trying to hit with the material. Diafiltration, and then quality control, so this is running some gels. So these sort of basic fundamentals are the same in both situations. Of course, the final fill in these two situations can be quite different in the sense that we've got on my left the research grade products sitting in the freezers there, and on the other side we have the final fill of a clinical product, which is under sterile conditions and in different types of vials. So what are some of the other differences? Scale and environment. So on the one side we have the research grade. Each one of those flasks could be a different product in a large room manufactured simultaneously. On the right side, this shows our 300-liter fermenter. You can see from the room and the gallooning of the operators is a very different operation here. We want to make sure the production is consistent from lot to lot, so there's a lot less variability and reduced carryover as we change over different products in the room. The other thing that is going to be different on the clinical side and commercial side is in-process QC. So we want to understand the process, in pretty good detail, so we know what's going on, so we can both make sure that we hit our goals for the manufacturing events, as well as ensure a consistent process manufacture over, different products, and the same product is produced in the consistent way when we do resupplies. The other difference is in the facilities themselves. So on the left side, we have large facilities. You can see multiple tips running simultaneously there, room with multiple operations going on at the same time. On the right side, very different. We do have multi-use facility, and that's really important as we try to support different products and more products coming through. So it's not just about making more of one product, but we have lots of companies out there, and hopefully, Teresa, when you work with your collaborators and are ready to go to the clinic, we'll have the ability to help you out with clinical production. But we want to be able to have more projects in the facility so we can support more programs. So it's designed to have up to 8 or 9 simultaneous products in the facility, based on how the air is handled and things like that. But to ensure a high-quality product that meets GMP standards, as opposed to research grade, where we're really trying to provide something with a quick turnaround for clients. And the reason the one side doesn't have anything on it is we don't do any really monitoring in the research grade facility, but those of you who took the tour last night hopefully saw at the end of the hall a large monitor with our building management system on there, right? So we've got redundant fans and redundant air systems, as well as a diesel generator that can power the whole building for, I think it's like 37 hours. We also have a backup power supply. Again, this is all to make sure that we can meet the deliverables and that everything is produced in a consistent manner and is safe. We do environmental monitoring on all of the GMP products that we make, so that's a key difference as you move more toward the bedside. And after being in the field for, I guess, going on 10 years now, it's really not a gene therapy talk until somebody puts up one of the Wiley pie charts. So, you know, the bar chart at the right just shows you the increase in programs coming down the pike. And this is, you know, very exciting, but it represents a challenge to us in the industry, which we've talked about, you know, at various times today. The pie charts talk about the phase, right? So if you look, phase 1 still the largest percentage of gene therapy clinical trials. Supporting those with various quality levels is very important to us. And then the indications, cancer is still a very large part of what gene therapy is attacking. And second, really monogenic diseases. We've heard a lot about that today, these rare diseases where we're approaching them with, you know, some sort of viral vector type technology to replace that gene. So, you know, we built this new facility and we're really ramping things up to be able to support these programs. So this is the pie chart in the vectors used. So adenovirus still used a lot, but we have even naked plasmids, and rAAV, as we all know, is becoming very, very popular. And this is, again, a figure that we've seen in different variations here to show about direct delivery or cell-based delivery for these products. And then what we like to say is Aldevron's inside here, in the sense that we help clients by manufacturing the core DNA that's part of these products. CRISPR is another area that we heard a lot about, especially yesterday. It's a very exciting field. We talked a little bit about Cas9 and rAAV delivering at least one, in this case 2 donor molecules, and we're privileged to be part of, again, these programs where we have the Aldevron inside, where we're making the DNA that becomes part of these viral vectors, as well as the Cas9 protein. So when you look at we've got those options, right? So we talked about, you know, the different manufacturing from research grade to clinical, and we have offerings in between with our GMP source. So what are some of the considerations on the path from the bench to the bedside? And I think it's important to keep in mind that these are just general considerations when you're looking at, you know, how to risk assess a manufacturing process and drug development so that you're doing the right thing for the right stage of development and for the patients at different points in the raw material and the supply chain. So we consider the complexity of the material, the stage of development, and the proximity to the patient. So complexity of the material, this is kind of just this sort of gradient. Sort of on one end, you have live cells, right? They're very complicated. You know, it's very difficult to get consistent manufacturing. We talked today about, you know, pulling cells out of patients for autologous type therapies and ensuring that consistency, or at least identifying it and be able to have assays that measure the variations that can occur. And then sort of on the other end of the spectrum, we have small molecules, which are, you know, very easy to characterize, and sort of you know what you have. And I think that, you know, nucleic acids tend to be more on that side of things, and that's actually a good thing, because as things are less complex and more easily characterized, you can have potentially more reliable manufacturing, which actually gives you flexibility that you may not have with more complex materials. We'll talk a little bit more about that. And then the stages of development. Of course, this is kind of an obvious one to everyone. As you move through, you know, lab studies through, you know, late phase and commercial, you need increased quality oversight, process control, environment, consistent manufacturing, the ability to scale up. But I think it's important to note that even through these different stages of development, there's still opportunities to make improvements, and I think the industry and even the regulatory agencies really expect us to do that. So then if you look at proximity to the patient. So in this case, we're starting with a cell bank, making plasmid, and the plasmid really is the API and is the drug that you're delivering to the patient. It could be in a liposome, hopefully in one of Teresa's polymers, or complexed to a gold nanoparticle. So this is the case where you sort of would imagine the highest quality because you're the closest to the patient. So when you look at some of those assays that I talked about before, the particle matter in injections and things like that, that's where clients tend to look at expanding that panel of quality control assays when they're getting close to the patient like this. Let me go back there. We have clients like Genprex that are in this camp where the DNA is the main component of the drug. And then here's a case where mRNA is the main component, so you can imagine that has to be a very high quality. It's going directly into the patient. Plasmid DNA and the enzymes used to make mRNA are one step behind that. So we can think about how do we optimize things to make material at that stage as a raw material that is high quality and sufficient, but yet is appropriate for its place in this sort of production supply. And it even goes back to the bacterial cell banks. How do we approach making improvements in the bacterial cell banks? If we come up with a better mousetrap, so to speak, what is required for us to make that change when you're making this raw material? And the opportunities to make those improvements, I think, are available, and we need to keep considering. Again, this is a case where, again, we're even farther away from the patient in the case where mRNA is transducing a cell, so there's even more opportunities, I think, to make improvements there as we develop the drug itself. And then this is probably one of the more common cases that we're working on with clients now is a case where the viral vector is delivered to the patient in situations like Luxturna. So the viral vector certainly is the API, has to be very high quality, full GMP. And there's been some discussion, as Henry brought up previously, about how to characterize plasmid in these cases, raw material versus process intermediate. You might say, well, gee, the mammalian cell bank is sort of at the same level there, and we know it has to be GMP, and people are very concerned about the lineage of the cell bank and things like that. But that's a much more complex material, so there's a lot more variation, a lot more things that you need to control there. So I would argue it makes sense to have that be GMP, but on the plasmid DNA side, I think as you move more toward commercialization, that will be GMP, but I think there are opportunities where you can make improvements throughout the development process and possibly not require a lot of comparability and things like that to implement those improvements. And then there's another case where even more ripe for improvement, where in the case of the CAR-Ts and whatnot, the plasmid DNA is yet another step removed from the patient. I think there's even some discussions here on what is the viral vector in this case. Is it an API? Is it a starting material? Clearly, the transduced cell is the actual drug. So I think we're going to see the thoughts evolve as the industry sees more products come down the line here. So this is, you don't have to read the whole statement from Scott Gottlieb, but I wanted to take out a part of this about the direction Congress gave the FDA by creating vehicles like the accelerated approval pathway is how do we get things faster, right? So we want to get this little guy across the finish line even faster so we can treat more patients. And Teresa had this slide up as well. This is some of the bottlenecks that we see. It's a virus shortage. We're experiencing increased demand for our products and services. So these are the roadblocks that I think we're all trying to get a handle on. And as we look at these different points in the supply chain, how do we make progress with that and really get things to patients faster? So this is Michael Connolly from Immunology. I don't know, Mikey, are you out there? He's one of my new favorite people. He presented a poster yesterday that I thought was really good. So I blew up part of it, but unfortunately you can't read some of the conclusions, so I typed them in there. So this is a functional evaluation of isoforms present in DNA vaccines. So what his conclusions were here, that supercoiled and open circular isoforms were shown to be competent at directing gene expression in vitro using both lipid-mediated transfection and electroporation. As other conclusions taken together, these results could be used to substantiate a reduced supercoiled specification for pDNA vaccines. Remember, before I talked a little bit about things that drive sort of the cost, and supercoiled percentages is certainly one of them. And I think that we need to look at some of these things, and just because somebody wrote down something, 2 or 3 years ago in an SOP that it had to be a certain percentage, what are some of the rational things that we can do, a risk-based assessment to ask the question, is that a good thing? Can we make improvements? We're not trying to cut corners. We're just trying to do a risk assessment of options based on sound scientific principles and data. So the other thing we need to do, and we are doing, I think the industry in general, is scaling up and increasing throughput. We talked about that a little bit before. On the bottom left, a bar graph shows the increase in manufacturing space for different CMOs in the area. So Brammer and Cobra and Urogentech and Paragon are all increasing their capacity to meet the demand, which is a great problem to have because it means that these things are actually working. The other area we're doing is we're scaling out. So these areas of personalized gene therapy that Lawrence Cooper talked about yesterday, you've got this therapeutic vaccine on a per patient basis. So we have patient 1 through N, and each patient gets their own DNA sequence. So what we have to do is generate a product for each patient. Very short turnaround time, on the order of weeks. So what does GMP even mean in that concept? And I think we in the industry are inventing these things as we go along and coming up with new ways to do things with parallel processing and closed systems that can support this and still maintain the safety and efficacy of what it is we're producing. The other thing that we've done in the past 12 to 18 months that's been very exciting is looked at what we've been doing for clients and saying, we do a good job at making custom products for clients. They give us the starting material, we give back to them more of it at a high quality for their use in their programs. But as we looked at it, we identified some areas where we can create some products off the shelf at these clinical grades to really enable our clients to move faster through some of these programs and to get them a consistent manufacturing product. So we have PLDX80, which we did in collaboration with AskBio. And the attitude for both Aldevron and Ask was not necessarily to make a bunch of money here. I like money. But it was really about enabling the industry. So this is a case where we've been able to offer a product to clients. They purchase from us, and there's no reach-through royalties. They can use it to make a billion-dollar drug, and they're good to go. And that gives them a lot of comfort in using some of these products, and it's a solved problem for them. Like, do I have to go find a helper? What helper to use? This helper's been around for 20 years. It's made material in dozens of clinical trials. And we're looking for other opportunities with these plasmids to offer off-the-shelf products. So we're talking to people, and certainly if anybody out there has got some ideas about products that we can offer off the shelf at clinical grade, we want to be able to pursue those to really help our clients. And on the Cas9 side, a little more complicated IP for sure, but this is a case where, again, we're trying to have a product off the shelf, consistently manufactured, available to ship to you the next day. And we, as we recently announced, expanded this to the SPY-5 through our collaboration with IDT. So this is really a great transformation of the company from a purely custom manufacturing organization to having products that we feel will really benefit the field. So we're hoping that's going to improve things, and we ourselves are continually improving. Jenny and her team and Henry are always looking for ways to do reasonable risk assessments on everything that we do to determine where are there opportunities for improvement, doing process simulations to show that we are doing the right things and that we're reducing carryover, we're implementing single-use equipment wherever possible, and really doing our diligence on these production processes so that we can ensure that we can meet these higher standards as products are moving toward patients and toward commercial release, as well as being able to get material to clients as quickly as possible and for reasonable costs. So I think, almost everybody has a story like this. So this comes from an email that Michael had received from a parent who had started this Indicure Foundation for Infantile Neuraxial Dystrophy. So I talked to her and another woman running the organization who said, we've got this disease, we know the gene, we've got some data, some research data, what do we do now? And your heart goes out to these people and you want to be helpful to them. And so we try to help as best we can and advise clients, even in areas outside of our sort of norm. Michael jokes, we'll make an F-16 for you if you think that'll help cure someone. It might take us a little while to do it, but we'll do it, or we'll find someone who can do it for you. So we just really want to be part of the solution for these patients and these children, and that's what we're all about and why we're trying to do these things better and faster. And this, I admit, this is a shameful plagiarism of some slides that Michael has given before, but I think it's such a good visual metaphor that it really it makes a point that this is where we are right now in the field, right? We're just in the Model T. We're all excited because we're not in a horse and buggy anymore, but it's still a Model T. So what we really need to be in this, so not that everyone can afford this, but this is certainly the type of car we want to be driving, because it's this type of car that's really going to make a difference to patients and difference to the field. So along those lines, kind of this is not an expected slide for me, but I was watching this show, The Greatest Showman, which is actually a really good movie if you haven't seen it. At the end of the movie, there's a quote where P.T. Barnum says, "The noblest art is that of making others happy." And although my guess is he probably didn't have biotechnology in mind when he said this, he was talking about more entertaining people. But when you think about it, when somebody's not suffering anymore, when their kid is now alive when they should have been dead, I would argue that makes them happy. So I think we are involved in the noblest art, and it's great to be part of this industry and part of Aldevron and everyone here in the field in general who has this noblest art at the heart of what they all want to accomplish. So thank you, and I'd be happy to take some questions.
Well, thank you, James. And I would invite everybody to come up and ask questions, since James was a very frequent requester of questions from other speakers. Now's your time to do a fair turnaround. Henry?
I'll fair turn around on James. Right. That'll be fun. Now, James, you are in a unique position, having worked for a lot of customers and now working at Aldevron. So to ask a question similar to that I asked this morning, sort of advice for folks who are just starting out, maybe admit some mistakes you've made in the past or things that people can learn from in terms of viral supply, DNA supply, just all along the development pathway. What do folks need to sort of look out for, or maybe a mistake that you would have improved on in your past?
That's great, and I'll put myself out there and throw numbers around. Whenever you hear people try and predict elections or stuff, everybody's afraid to put sort of definitive. I would say, one piece of advice is the day you decide you need viral vector, it's 2 years until you're probably going to get it. So that includes, getting the plasmid made and everything like that. So that's if you know what the sequence is and you have data and all that stuff. That's what I would tell someone. That's what I told the woman on the phone, when I was talking at that patient organization. So I think that's something to keep in mind. It's going to be that, because we heard people say they're booked up for 2019. So that's one piece of advice that I would say. I think the others would be, when I was at REGENXBIO, it was just the number of assays and the amount of characterization you're going to have to do with viral vectors, and especially the cell lines. Plasmid less so, but there's always some other hoop to jump through that you didn't see coming. So, and that's part of what that 2-year period will be, is allowing for those hoops that you didn't see come up and being able to have time to jump through them. So I think that, just be realistic about how long it's going to take you to get from point A to point B, and talk to people in the industry. So, 10 years ago, there wasn't a lot of gene therapy going on, and there wasn't certainly a lot of industrialization and things like that. We have conferences like this. ASGCT is a great one. Facilitates meeting in January is a great one. Get out there, talk to people, join industry groups. There's a bioforum operations groups that we recently joined that has a cell and gene therapy section. Talk with your peers and learn from them so that you can be as efficient as possible.
Okay, next question.
Yeah, so I've had the pleasure to work with Aldevron and see its growth and changes over, the past several years. And I just was wondering if you could talk a little bit more about the future of Aldevron. I mean, you guys have scaled so much, the past couple of years, and really the next direction, what does the next year look like, the next 5 years? We all took a tour of Building 1 yesterday, and just if you could talk a little bit more about that and any new directions that you could envision Aldevron moving into.
Now that really puts me on the spot. And I'll ask Jenny to help me with that one too. As Jenny referred to, that's Building 1, there's going to be Building 2. So I think for us, growth and consolidation. So we have operations in other locations. That's not the most efficient way to do things. And I think Michael's vision is to bring those things together. I think that the other thing we talked about products is, Michael and John's vision is to do a good job at what we do, but really look forward to our clients to see how can we help them even more with things like these off-the-shelf products, with better products. I think we're in the unique position, as people have said, where we see lots of different things going on. So I think we've hit that kind of inflection point where we have seen enough that we think we can be even more helpful to people. And when it comes to, plasmid manufacturing, Cas9 manufacturing, we're looking at ways to, the constructs and improving their, let's call it manufacturability, and what things we can do to make our yields higher and things like that. And that's why when you look at some of those supply chains and stuff, we can't be afraid to get better just because something's sort of locked down, so to speak, because you're in phase 1 and things like that. We're being proactive and reaching out to regulators and saying, okay, and I did this. I said, here's the bacterial cell bank. It's way in the front of this thing. If we have a better cell bank or a better set of conditions to grow the plasmid, which is a raw material for something else, can we make that change without requiring clients to do a bunch of comparability stuff? And I think the answer is going to be yes. It depends on all of those things, how close you are to the patient and everything, but I think, we want to continue to innovate in these areas, and I think there are opportunities that are ripe for it, and I think it should be the expectation. We shouldn't just say, okay, we're in the clinic there, so we're not going to worry about making anything better. We always need to make these things better. So I think those are the opportunities that, at least, that Michael and John has put out for us to improve and make a bigger impact on the industry.
Yeah, I would just add to that, I guess, from like the operational standpoint. So in the time I've been at Aldevron, we've always talked about how it's a biological process. It takes X amount of time to be able to, have a fermentation growth complete or things like that. Our next phase in 2019 is going to more of that, like Henry talked about, where, it's kind of scary. We're being compared to, with standards as an automotive facility. But however, in the same token, we're going to start going to that shift work so that we are doing more of that continual. And then at that point, the biological process gets taken out of that equation. It's still there, but if you have multiple shifts going, then if it ends at 3 in the morning, if that's a normal shift, then it's really no different than if it finishes at noon. So we'll be looking at things like that for our expansion as well, so that we can get even more turnaround. Like James said, we can be doing 9 products at one time in our GMP facility, but if you're doing that rapid turnover, those 9 products per week then have a greater impact. Any other questions?